microscopes and how we use them to draw biological images. So I'll run through a few things. We're going to start with two kinds of microscopes. The one on the left is a microscope. The one on the right is a scanning electron microscope. Now, you also have transmission electron microscopes, and we'll go into the difference between the two in a second. This is what you're probably more familiar with. They're a lot cheaper than an electron microscope, and you have a little bit of light that comes here. This when I was at school and we had these. We had a mirror, a little lamp here. We'd have to angle the mirror to shoot the light up. You've got an objective lens and you've got an eyepiece lens. To find the magnification, you multiply the objective by the eyepiece. So let's say that's 40 times and that's 10 times. You'd have a 400 times magnification. And you prepare your slide normally by making it really thin and adding a stain like iodine solution to make it a bit clearer. Put it on the stage. Use the focuses here, the focus wheel here, to move the stage up and down. And as a result, that will focus the image, give you a clear image. And you can actually now take photos down the objective lens with your phone. Now, these are quite straightforward to use, right? Now, these are a lot more difficult. We don't have them in school. Now, they work on a very different basis. You have a sample. Now, that sample is kept in a vacuum. Now, the reason it's kept in a vacuum is this uses electrons instead of light. So, whereas light has sort of a wavelength like this, electrons just go in a straight line, which means that they can easily bounce off air particles, which should distort your image. So, this is kept in a vacuum, and we fire electrons down, and then they bounce off in a scanning electron microscope, and they go through in a transmission electron microscope, and the image is picked up, and then they computer generate an image. You don't look through an eyepiece lens for this. Now, in this case, in a scanning electron microscope, what you'll do is you get your sample and you'll coat it in gold or another metal, which will cause the electrons, because of the outer electrons, the C of D localized electrons on a metal, it will cause the electrons to bounce off, and the angle it bounces off, the computer can figure out the image. Now, there is actually a little gold water bear at my university that water bears are the only animal that are able to survive a vacuum, and one of them just got up after being put through a scanning electron microscope and just walked off, and it was already coated in gold. So they're very resilient sort of things. Now, these have got much higher magnification and a much better resolution. And by resolution, we're talking about how clear it is. Because if I've got an object, we'll have a look at this in a second. A light microscope has a wave, which means that everything within that wave is blurred. An electron microscope just goes through, which means it doesn't blur and it doesn't overlap the images. Now, the difference between the two is this. This is what you'd get in a light microscope. These are onion cells, and this is an animal cell that you'd get in a transmission electron microscope. And you'll see that there's a lot more detail in this. This is actually what this looks like, the inside of this. It will have these little bits and bobs all the way through it, but you can't see them in there, and that's because of the resolution. So the resolution of this image is much better, and you can see all the different little membranes in it, and that's because, as I said, the electron goes in a straight line, whereas in a light microscope you get a wave. So therefore there's a higher there's a worse resolution in the light microscope. This is also a lot closer. So this is one cell, whereas these are lots of cells, right? And that's because the electron microscope, transmission electron microscope, has got a higher magnification as well. So now, if we get these, and you get an image, and you're asked to draw it as a biological diagram, what you're going to do is you don't sketch it, you don't colour it, and you don't break the lines. And you must have the image that you draw be big enough to fill up over half the space the exam gives you, but also it needs to resemble the image you've got. So what you're looking for on your diagram is a line. Let's change the colour just to make it a little bit clearer. We could also make it a bit thicker. Here we go. So your image will have a shape like that, and then a shape sort of a bit jagged around, and you might sort of put in some lines like that. Now it must, things like that you would ignore because that's kind of just a bit of shading. So if I was to draw this as a biological image, what the exam board would be looking for is something that looks a little bit like that. So don't overcomplicate these images. They might expect you to draw the cell membranes. Don't overcomplicate the images. They're looking for very simple images, no broken lines, single lines, no sketching, no colouring except for a nucleus.